Okay, you guys ready to start? Oh, this is gonna be tough because we got a lot of people in here and it's really warm. Happy New Year. So this is coming up. So hopefully you're in the right place. If not, now's the time to get up and run out, right? In case you're in the wrong way. So my name is John Cartel. If you don't know me, I am a molecular biologist, so this is a perfect fit for me because this class is basically my baby. I've been teaching it for, wow, 12 years here at SUU now, 14 years overall. So it's, I love this course basically because this course is, you know, the nitty gritty of the cell. Everything you wanted to know, well, not everything. A lot of the things you wanted to know goes on in the cell at the molecular level. That's what it's all about. So that's why I love it so much, because every time I teach it, it's just so exciting looking at the complexity of a cell and everything that goes on there, just how amazing that is. So it just blows my mind every year. And every year we learn new things as well, new stuff published. So constantly adding things to the course. Okay. So today we're going to do a couple things, of course, talk about the course. I'm not going to read through the syllabus because that's really boring. I suggest you do that though, because there are some, well, what do we call them? Rules, guidelines, pirate bylaws, what do they call it? The pirate code, is that what it is? We've got guidelines, right? Things like makeup policy, learning objectives, that kind of stuff that you probably should know. So you want to go back and read those things because there are some, we'll talk probably a little bit more about it, but I'm kind of a stickler with my deadlines and that kind of thing, especially with a course this large. We have almost 70 students in here, and that's a lot of grading with all the things that I have. Yeah, it's pretty big. So that's kind of the idea, and also to get you psyched up, right? Because the course is awesome, and I want you to like it, and of course engage with it. The worst thing that could happen is you're here because somebody's forcing you to be here. That's just going to make this course a drag, because it's kind of like a boot camp, right? If you're forced to go to boot camp, that's not always the most ideal situation. I kind of describe this course as a boot camp because we do a ton of things. We go through a lot of material. We read a lot of scientific literature. So if you're still scared of reading scientific papers, it's going to be a little bit of a struggle, but I want you to get better at it. So that's why we're going to do it, like 8 or 9 different papers we're going to read over the course of the semester. We do some writing, you know, everyone needs practice writing. Okay. I don't care how good you are. But if you're a senior, especially, you shouldn't be graduating out of here unless you're able to communicate at least satisfactorily in science, right? So we practice reading, we practice writing, because we all need that. And of course, we look at what's going on in the cell. I was just thinking about it this morning, kind of how I describe this to everybody. So when I teach Gen Bio, which I do every semester, I always talk about how it's the tip of the iceberg, right? Gen Bio is like that. You've heard of that term, right? The tip of the iceberg. And what does that mean? Right. There's a lot more to that iceberg, right? Just think of, oh wow, what's the ship? Titanic. Wow. Okay, Titanic, right? So that's kind of where molecular biology is, right? And you probably think back to Gen Bio, especially if you're in my class, and you're like, really? That was just the tip? Yeah, because this right here then is molecular biology, really, is the rest of the detail that we kind of left out from Gen Bio. And that's really what we're going to do in this class. We're going to focus on things like regulation, dysfunction. So I kind of start out, and this is the tough part. I expect you to kind of remember your Gen Bio and your genetics, right? You probably saw the same stuff in genetics. Things like transcription, translation, replication. You need to go back and review that because I'm not actually going to go through the detailed mechanisms again. You should have gotten them 2 times by now, hopefully, depending on your professor, okay? So, but my expectation now, though, is that you could go back, review that, and most of that will come back to you. Okay, I'm not going to really grade you on the nitty-gritty there. But then what I want you to be able to do is kind of apply it to this. And so really this is kind of the wrong picture, isn't it? Because what does Gen Bio do? What is it supposed to do for you? Right, it's supposed to be the foundation. So I don't know if it makes any sense to write it like this, does it? Not really. It's the same idea, I guess, right? You have gen bio, genetics kind of give you the foundation, and then your upper division stuff is up here, isn't it? Does that make any sense to you? Or we could draw it as just the foundation for a house or something, right? You know what the foundation is, okay? So that's kind of what I think about this class, is that we have a foundation, hopefully, and I know it's tough because you may not have reviewed this stuff for a while, so it might take a little bit to come back, and that's okay. But once you kind of regain that foundation, now we can dive much deeper into all the cool things that go on with that. So not just looking at like, okay, transcription, right? That's just, should I start picking on you right away? Anybody remember what transcription is? You know, I know. Oh, Christ. No, transcription is DNA to RNA. Right? So you learn that in gen bio and say, okay, good RNA. Okay. Did you know there's like 4 or 5 steps in that one process? So that's what we're going to do. We're going to dive into transcription and look at, not just pre-transcriptional regulation, but there's also post-transcriptional regulation and splicing patterns and other modifications. There's a whole RNA code, just like epigenetics, they call that with DNA. DNA can be modified in other ways than just the sequence. That changes how you use it. Same thing with RNA. There's a whole separate RNA code. Okay? So that's kind of what we do in this class, is to dive into that nitty gritty and just really look at how complex it is. And then, of course, if you're pre-med, you're really interested in this, aren't you? Or not just pre-med, pre-vet, whatever. If you're interested in what goes wrong with the cell, yeah. Well, we're going to see that a lot can go wrong. And when it does, that's going to have an effect on the cell, obviously, because these processes, as it turns out, they are carefully regulated. Carefully regulated. And when things start going bad, what happens? What can happen? Cancer, death, cell dysfunction. If you just think about cancer, we'll end it. This is like at the end of the semester. But cancer is, it does the opposite of what every other normal cell does. It doesn't listen to anybody. It does its own thing. Its genome is totally messed up. If you've ever looked at one of those HeLa cells in a karyotype, it looks nothing like a regular cell, and yet it grows just fine, doesn't it? Which is bad for the host, usually, right? Yeah, so it's totally weird that a cancer cell can be so messed up and yet so well functioning, if you excuse the expression. Okay, so that's kind of what this class is about. We get to dive in and start applying this stuff and understanding what's going on and seeing a lot of stuff we still don't understand. Still some processes we don't have any understanding. Okay, and as we do that, we're going to get practice reading and writing and extracting information from scientific literature. Because if you look at a textbook, where did we get all that information? Yeah, right. Research. So they didn't just sit down and write it from scratch. They sat down and wrote it from other people's research. Okay, so that's kind of what we're going to do. We're also going to get a good helping of molecular techniques. So in the lab, we're going to go through a bunch of common techniques that we use to modify genomes and to splice things together. You have to do the evil scientist lab. That's part of the requirement. You have to perfect your evil scientist cackle, right? That's good. Okay, that's a different class. Maniacal laughter 101, I think. So that's what the lab is. You're going to kind of do your own little research, actually, your own little project on transcriptional regulation. It's kind of an interesting project where we do some cloning and then test transcriptional regulation. So it's kind of a fun part of the course. Okay, so that's kind of the idea. I really think of this almost like a capstone. You're almost at the top of your level now, where you can take all your science and start engaging with it and applying it and understanding it to a whole nother level. And that's kind of what we do in this one. And it is a weird class. It's cellular and molecular. So toward the end, we'll get a little bit of what they call the classic cell biology stuff, where you look at cytoskeleton or cell cycle regulation, that kind of stuff. So more protein regulation, but they kind of go hand in hand. So it's fine to do them together. Okay, cool. So you excited? Did I get you right up now? This class is awesome. Of course, I'm biased, but I think it's great because we do get to look at a lot of the nitty gritty there. Okay, so what do we have that's going to help you? So we can go through this. Obviously, hopefully you're a fan of Canvas. Put a lot of information in Canvas. Okay, so you should go through and check out here. I've tried to help you out with a lot of background material. Okay, so if you need to go back and refresh yourself on things like cell structure or macromolecules, do it. Go back and read about them. Tomorrow in lab, we'll be working on this GenBio review, which is just a set of old test questions that I use in GenBio. And the purpose there is just to try jogging your memory. Okay, so hopefully you've memorized it at some point. We just have to get it back out of your brain. And some of us will have to review a little bit more than others. That's okay. Some of us will see areas that were totally skipped in our class or something like that. And so this gives you a chance to start going back and reviewing that. Because again, when we start off here with DNA structure, I'm just going to kind of dive right into it and start talking about the cool aspects of DNA. That'll be on Wednesday. And when we start DNA replication, I expect you to kind of remember the basic steps. So you have to go back and kind of review those enzymes because I won't be spending much time on that. Okay, and I do that on purpose. I don't really want to have to repeat it again and again and again for the third time, really. So hopefully you can do that. Do a quick review and get yourself caught back up. Okay, and if you need help, you have this. You have me. You have the tutoring center. You have fellow students. So you should be joining study groups. We'll form lab groups in labs tomorrow. So that's a natural starting point for study groups. Yes, ma'am.
I was looking at your syllabus earlier. Don't your office hours listed?
Oh, wow. I totally just forgot that. Yeah, so it's listed on my door if you want to go by my office. Otherwise, I'll try to update that. So yeah, somebody else said I might have the room wrong on there as well. So I'm going to have to go back and check my syllabus. Sorry. There's always something I forget or screw up on, and you guys just totally need to call me out on it because that helps me remember what to do. Or send me an email with a lot of expletives or something like that. That's my to-do list, is my email, okay? Oh, and by the way, that's the best way to contact me is email, not the Canvas system, okay? And unfortunately, you have to put my full name in there because I didn't shorten it. But this is the best way to contact me. It goes right to my phone, right? Pretty much everybody's probably the same. If you try to message me through Canvas, I might not see it for a day or two until the next time I log into Canvas, okay? So don't, I will respond eventually. So try not to use the Canvas system. Try to just use the email system, okay? That's the quickest way to let me know what's going on. And you should, if you're sick, if you have an emergency, if you're confused, whatever, that's the best way to contact me, okay? So just shoot me an email when you see something wrong or a due date is suspicious or, whatever is going wrong, just let me know. Yeah, don't be afraid to do that. I'm still human and I make a lot of mistakes, right? If you guys are perfect, good for you. Good for you, okay? I've never been perfect, so anyways, there's a lot of background material here. There's helpful things like how to read a scientific article. Oh, that's nice. Okay. So maybe read through that, gives you some pointers on how to approach articles. Our first one will be the Watson-Crick paper on Friday. And that's a nice softie to start with. Okay. 2 pages, published in the 50s. They didn't really get into much detail. So a very easy one to do it. There'll be much harder papers filled with jargon that's going to be tougher. So we start off a little easier. So look through there. There's also makeup contracts. So I'm really big on this. If you already know you're going to be gone, I don't care if it's an athlete or a student ambassador or whatever. I don't really care why you're going to be gone. Look at the schedule. If you know you're going to be gone, see what you're going to be missing. Come up with a plan to take care of it before you go. Okay? So just take a look, right? Take the next couple of weeks. Look at the entire semester. Put our exam dates probably won't change. So put them in there. Remember, I've been teaching this long enough. Things don't really change much in the course, okay? So put stuff in there you know you're going to miss. Come up with a plan and then come talk to me, okay? So I know you have a plan to either make it up or whatever you need to do. And the class schedule is here as well as in the syllabus. So if you click on that, it shows you our daily topics, shows you our lab topics. So you should know what to read before coming in the lab. It shows you where our articles are. So for next week, you're going to have this short little analysis that's due before we discuss it in class. So it's a little harder there, okay? So I actually expect you to try to read the papers, extract information from it, and then we go over it in class, because I want to see your interpretations first before you hear everybody else's, okay? So you can kind of see and expect what's coming up, and you can be prepared. You can start looking things over, okay? So I spent a lot of time in Canvas trying to put stuff there to help you in the modules. So again, you have backup material, you have old audio files. So these are getting a little dated, but most of the material should be the same. So if you have I think it's a Dropbox, you can actually get in there and download all those audio files to your own Dropbox. And then if you need to come back and kind of listen to the notes again, then sometimes that's helpful if people are listening. What do they call it? Auditory learners. Because remember, you all kind of are different styles of learning. So I try to put out resources that you can use, but it's really you that knows how you learn best, right? So whether it's memorization, I always recommend flashcards, but that may not work for you. There's probably Quizlets or something, right? Those are helpful as well. I am going to be doing videos, but this is going to be mainly for people who are sick or have an emergency. So I will share videos, lecture videos on a limited basis. I really do expect you to show up as much as you can, okay? Now, if you're really sick, please don't come here, okay? Nobody wants to catch whatever you have, okay? So if you have an emergency, if you have a sickness, I will work with you. I am flexible in some circumstances, but for the most part, I expect you to be here, okay? Just like I'm here every day if I can. So I expect you to be here. It's just like a job, right? So there's audio files. I left the study guides up from last year. So they're mostly the same. You can kind of see what kind of topics I'll be testing on. The tests are all written. They're not multiple choice. So they're all short answer type questions, okay? So I'm going to be, telling you to take the material that we learned and apply it to a new situation, or synthesize something new from something that we've talked about in regulation. It's remember what we're talking about here? I mean, look at this. Is this lower level learning or higher level learning in this class? Think about Bloom's taxonomy. Have you ever heard of that? Well, I feel like I'm talking a lot. I am Californian, so born and raised in California. So I do kind of just go on and on and on, which is good to ask me questions, right? It's excellent that you raised your hand in class. That slows me down. It's good. That's a good thing. And I know it's going to be a little scary to ask questions in a class this big, but again, you guys are juniors and seniors. I hope that you figured out a little bit of college already, yeah? So hopefully you've been through this a few times. You can get over those stuff here. If not, write your questions down and come ask me later, right? So, what was I talking about? Have you heard of Bloom's taxonomy? What are the lowest levels of learning? Memorization, regurgitation, right? Memorize something, spit it out on a test. It probably won't work in this class. It probably doesn't work in my gen bio class. Okay? You have to really understand it. So you look at the higher levels of learning, it's like synthesis, synthesizing new information, analysis, analyzing all the stuff. Okay? So that's what is really going to be expected in these exams. I'm going to be expecting you to take the information we use, create something new, and then come out with an answer, or like give you a different situation. Use your knowledge to answer that. That's kind of what employers look for, I think. They look for people who can adapt, who can synthesize things, can use that information, not just try to memorize it and then not really understand what they're using. Okay, so it's a little tougher. But I think this is why I get such strong reaction from students. I push you. I tend to push you to be better, right? I want you to be better. I want you to graduate and go and make us proud and cure cancer and all that that stuff, okay? And sometimes students either love that or hate that, right? So I kind of get polar reactions sometimes from my classes, okay? Which is, I'm fine with, okay? Okay, so again, look through here. Unfortunately, a lot of our classes will be lectured. It's still biology, so I have to lecture. You can see the lecture notes that I'll use are shown here in PDF. This is a paper. Those are the lecture notes. We will be using a system though called Top Hat. So I sent out an email this morning to whatever email is in your Canvas account. So if you didn't get it, go check that email you never checked for school, or check your spam folder. Sometimes 50-50 chance it goes there, okay? But you should have gotten an invite from Top Hat. Did anybody get it? Okay, good. So it's working for most of you. If you didn't get it or you don't use that email anymore, send me an email. Say, please send me my Top Hat invite to this new email, okay? And then go change it in your, not Canvas, but it'd be my SUU, right? Go change your email address, because that's where I get all your email addresses from, okay? So if you didn't get it, let me know. If you did get it, you should go in there. The Top Hat system is pretty good. I think it's a good alternative to a textbook. It's cheaper than most textbooks. What it also allows me to do is give you an ebook as well. So there's some ebook chapters in there. So you can go in like this one. I think it should be visible now. You can go back and review your cell molec, chemical bonds, biochemical, whatever. And then when we get into, say, nucleic acids, what I'll do is I'll present those slides during class, and then I'll pepper questions throughout. It, okay? And so, sorry. This is how you're going to get your attendance credit, is by being here and participating through Top Hat. So another carrot to make you come to class, I guess. Also, so you get points for that, but also it's helping you engage with the material. So you're not just here, sitting here listening, trying to learn by osmosis. I'm asking you questions, and sometimes you have to do pair and share or something like that. But I'm trying to get you to think about higher order kind of ideas, which, you know, might be on the test, right? Hint, hint. Okay, those kind of higher order questions. But that's what I kind of use Top Hat for, is to help you more engage with the material during class. Again, most of your learning is probably going to be outside of class, right? How much class time do we spend together? 3 hours. Less than 3 hours a week, right? Because it's only 50 minutes. So most of your studying and learning is outside of class, isn't it? Well, you probably already figured that out. And for a class like biology, it's usually more. So they say like 3 times the amount of time outside of class. So that's 9 hours, okay, of studying a week. So hopefully, you know, this is a different pep talk from when you were a freshman, right? As a freshman, you didn't really understand what you're getting into. Hopefully now you understand. This class is a lot of work. It is a lot of work. So you have to be ready for that, okay? So anyways, you'll find some homework in here. So those due dates, I think, are also listed in Canvas, but you'll be completing it through Top Hat. So it's basically reading through the Top Hat chapters and answering questions on it. It's not too hard. And then when you're in lecture, you'll get points for participating. So you want to make sure your phones or tablets are attached to the Wi-Fi before Wednesday, because that's when I'm going to start using Top Hat. And the cell signal down here is bad. That's why we have this little phone right here, okay? So you want to make sure you can use some sort of device, either a tablet or a computer or whatever, during class to participate with that. So, any questions on Top Hat? And it's not perfect, but I think it's a nice little alternative to buying a really expensive textbook. Did anybody buy it already? It's only like 66. So probably half what a used textbook goes for. No, maybe a new textbook goes for. So I know it's money, but I think it's well spent, and it helps you engage with the material. So No questions on that? 66 bucks. And don't buy it through the textbook store because they mark it up, don't they? So buy it through the, don't say it, don't tell them I said that. But they buy it through the website. It's cheaper usually. Now, okay, so we have top hat there. Of course, we have Canvas. You'll also find the papers listed here as well. So you can look through, and this is kind of set as we go through it. So I call our first section DNA metabolism. So don't think of metabolism as digestion. Think of it as how do we use DNA, basically. How does the cell use DNA? So we're going to look, of course, DNA structure, chromosome structure, replication. How do we change DNA through recombinant DNA? How does it change itself? Like mutation, transposition, genes and genomes, those kind of things. So the first section is all on DNA. It's kind of how I put it together. And then you'll see we move on. So the next section then is pathways. Oh man, how huge are pathways in molecular biology? Huge, right? I tell you in Gen Bio, cells don't do things by themselves. That's cancer cells. Most cells, they don't do anything unless told to, like the military, okay? They don't divide unless they're told to. They don't differentiate unless they're told to. They don't die unless they're told to. Okay? Right? So pathways are critical in this course. We really have to understand how proteins are used to control processes in the cell. And I'll kind of give you a little example today. I think we should have enough time. So go through Canvas, click on these things. You should be able to follow kind of the schedule through here. Okay. I try to give you lots of things to use. And again, you can always come in and ask for help as well. Okay. So try not to be scared of me. I know I look scary. For some reason, my office is scary. I literally get nobody in my office. I have no idea why. But remember, I know what's going to be on the exam, right? It's going to be your best resource. And contrary to popular belief, you're never marked down for coming into the office. Never. I've never done it. Okay. So I try to help as much as I can, but it's best to come in with questions, right? Don't just come in and say, please lecture me again on this. I probably won't do that. But I can kind of help you through whatever question. That sounds fair to you? Okay. Sat in the front, man. I'm gonna pick up. It's okay, you can move on. I don't have a seating chart, so you guys can move around. Okay, so questions on Canvas. Do you know where to find things? You have announcements. Oh, by the way, there's a couple things you have to do. Did I send out I didn't send something to you guys. There are some old announcements you may want to go back and review, but there are a couple assignments you have to do right away. What's it called? Attendance quiz. That is required by the university for, I think it's for scholarship requirements. They're making sure you're here. There's also a safety quiz for lab. So pretty much all of you should be enrolled in lab unless you've taken it before. It is a co-requisite. So it's expected they're both taken together. So there's a safety quiz for that you can take. There's something else. Oh yeah, the pre-class assessment. So this is kind of how I see if you actually learn anything in my class. So you can take this. This does not count against your grade. Okay, so just try your best. Don't use any material for it. And then what we do at the end of the semester is I give you a post-class assessment. And I basically show my bosses, I compare them together. And I see how little they remember it at the beginning, but look at what they learned afterwards. It's usually huge, a huge gain. Okay. So that one, none of them count against your grades. Okay. So you can just take them and get them taken care of. So anything on Canvas, you can also follow your grades. The exams do weigh heavily in the course. So you want to make sure you get help early and often, okay? And make sure you're ready for those exams because I want you to sound smart. I want you to talk and use the words and know what you're talking about when you answer those questions, okay? So they can be kind of tough. And I do have quizzes as well. So there's lower stakes quizzes that we'll take. These are through Canvas. I would suggest you take them on the honor system. So don't use any sources. I put a note in there. A lot of these, sometimes tablets don't show the pictures. So try to use a computer, try to use a laptop or a desktop. They seem to show those pictures better. But anyways, the quizzes are there to they're about a week before the exams. So they kind of gauge how you're doing. And if you really struggle on a quiz, then you have another week to prepare and get help, right? So I try to give you a lot of resources that you can use to help you out there. And there's the syllabus. I have to go back and change it, and I'll put my office hours in there. Sorry, I didn't do that. So any questions on Canvas? You guys pretty good with Canvas by now? And I'm sorry it's so warm in here. Whenever I ask the facilities people to change the temperature, they look at me dumbfounded. Like, you actually want us to control the temperature in the classrooms or in my office when it's freezing cold? I said, yes, I do. Yes, sir.
Are the quizzes short answer as well?
Yeah, I think the quizzes are mostly short answer, maybe some multiple choice on those. Yeah. They're usually like half an hour quizzes or something like that. I think is what I did. If I remember. Yeah, 30 minutes. And then it tells you the topics in here as well. It tells you the topics. So again, try lower stakes to try to help you start.
So I'm looking at it, I don't know if I'm just missing it somewhere, but I can't find the pre-assessment anywhere. I can see the post-assessment. It's not there.
Oh, okay. Maybe it's not open. I'll go back and check it. Is it under quizzes? Go to quizzes. It's under here, surveys. It looks like it's published, so I don't know why it's not.
I was looking under assignment instead of under.
Not showing up under assignments. Did I change the date? Yeah, it looks like I changed the date. Oh, available until. I'll go back and check it and see if there's anything I can change to make it more visible. If you miss it, it's really not the end of the world, so don't worry too much. I want you to take it though, because I want to be able to do that comparison. Okay, so we talked about Top Hat. We talked about Canvas. Talked about, again, you can go back through the syllabus that has the rules in there. Some of the things there. You can also look at things like the mission. I think I have the mission of molecular biology, kind of why I think it's important, what I want you to learn, that kind of stuff. A lot of good stuff on here. Okay, what else? What else are we talking about? So you can start looking at Watson and Crick. So that would be this one right here. It's a little different for Friday because you're not going to write up a synopsis. You're just going to answer a bunch of questions. And some of it's actually a lot of chemistry, right? You know that chemists were the ones who found DNA structure. So for you chemistry majors, this is my plug for you guys, right? Okay. There's a lot of chemistry entwined with biology, obviously. But that's pretty easy. And actually on Friday, what we're going to do is I'll just kind of start you off, but I'm actually going to have to leave class early because I have an NSF grant workshop meeting that I have to go to on Zoom. Okay. Starts at 10:30. So Friday, I'll start you off there and then you guys can kind of work together in groups to answer those. And then of course, next Monday, we have a holiday already. So Martin Luther King Jr. Day, right? So we won't have class on next Monday. So that gives you a little bit more time to get ready, right? Okay, yes, sir.
I noticed you had a lot of like papers in the assignment.
Yeah.
Are those like we're reading scientific papers, or are those like writing our own papers, or just kind of what's the idea?
Yeah, so these are the scientific papers that I mentioned earlier. So they're kind of scattered throughout. And my expectation here, except for Watson and Crick, all the other ones, what you'll need to do is you need to read it, try to extract kind of the certain information from it. So what is it about? What are the authors trying to tell us? Do they do a good job or not? And then you write kind of a short synopsis for it before we talk about it in class. Okay. So it's a little tough, right? If you're not used to reading the papers, there's going to be a lot of jargon in it, right? Just like any scientific journal. But that's okay. I think that by struggling with some of these things, it actually helps us learn it a little bit better. You know, I'm not just going to be, a lawnmower professor where I mow all obstacles down for you in life. You're going to have to struggle a little bit, but I think you'll gain from that. But then we'll talk about it in class, right? So we'll go over it, talk about the main points, why it was important, why I chose it, that kind of stuff. But those are those papers. One thing I didn't mention is you're also going to write a short little grant as well. Okay, so this is more writing. I know more writing, but here what you're going to do is choose a topic of your choice that you want to do a fictitious research project on. So you're not actually going to do the research. It can be on something you're working on right now. If you're doing research for another professor, that's fine. We can talk about this more later on. I want you to start thinking about your topics now over the next couple of weeks. You can come to me if you have any questions. But this is also something you're going to write up, and it's almost like an NSF grant. So you're going to put together some background material that's already been published, and then what you're going to do to extend that research. And that's kind of how you get money in this world, right? You say, well, here's what's been done, here's what my lab is going to do, and why we're going to do it, and give us money to do that. That's kind of what that's about. Okay. So I didn't mention that before. But the papers, some of them are harder than others. Some of them are harder than others. But I think a lot of them are cool in one way or another. That's why I chose them. Okay. And really the only way to get good at reading scientific literature is to read it. So remember, there's a thing back in the modules that helps you give you some ideas. Things that I do sometimes is I start with the figures, or maybe I just read the abstract and then look at the figures and see what are the authors trying to show. And then if you don't really understand a figure, then you can go back to the regular text and try to extract information from that. Sometimes even if I hit something, like a term or something I don't understand, what do? Just Google it, don't you? You just go to Google, or even Wikipedia sometimes helps out. And you try to see, what are they doing there? And then I'm like, oh, okay. So they're trying to look at RNA or something. And then you go back to the paper. That's kind of how I break it up sometimes. So it's tough. It's tough. You can do it. Okay. Let's look at some cool stuff. So why do we care? It's kind of the question. I hate this question, by the way. I get that in Gen Bio a lot. Why do I care about this material? Oh, man. So I really want you to want to be here, to want to learn this stuff. And so I put together just some intro. You can look at this in the cell molec intro, and we're not going to do top out today. But if you look at some of this material that I put in here, it kind of gives you some interesting things. If you think about regulation, right? I had it on here, regulation of the genome. You know, do all our cells have the same DNA? For the most part? Yes. With a few exceptions, right? Like sex cells or red blood cells. Every other cell has the same exact DNA. Do you know that? Well, what makes a liver cell different from a kidney cell, different from a skeletal muscle cell? Right. Where do you get the proteins? From the DNA, right? So that's really what molecular biology is. How a cell uses the DNA differently from other cells, which gives a rise to different proteins, and not just a couple. I mean, there's some estimates that cells have over 100,000 different proteins. From 20,000 genes? How does that work? I won't answer that. You have to think about it. How do we get all those proteins? So if you think about some organisms, how they change, really it's just changes in gene expression, right? Or the regulation of gene expression. So I have an example in here, like the Arctic fox. What happens to the Arctic fox? It's brown or red in the summer. And then what color is it in winter? White. Again, you probably already know, okay, survival of the fittest, right? Okay, something like that. But how does that actually happen? It's at the cellular level, isn't it? The hair follicle cells change the proteins they're making. Fur is this protein, right? So they change the protein they're making or how they make them, and that changes the color of it. So really that's what this class is all about, is looking at changes there. Another example that I use, if you ever take a developmental biology class, they love looking at things like the fruit fly. So the fruit fly goes through this carefully coordinated life cycle where it just starts from a fertilized egg, kind of like what we do, right? Our eggs are fertilized, and then they split into 3 different what is it, exoderm, whatever. I'm not an anatomist, so I don't remember. But here, same thing here. This tiny little fruit fly goes through these changes where it actually has different segments of its body, of its pupa, basically. And each of those segments then, at the cell level, are being driven to differentiate into a different part of the fruit fly. This is just a little fruit fly, right? Little insignificant thing. It's flying around your bananas in the springtime, isn't it? Or summertime in Cedar City. Whenever I squash them, I go, oh, there goes the cure for cancer. Or something like that. Because they're you know what they're called, a model organism, right? Have you heard of that term before? Yeah, so those are organisms we study. Why? To understand the gene expression there and then apply it to humans, because that's what we really care about, is humans. But anyways, look at this. If we look at how it actually does this, what are there? There's a bunch of genes that are carefully regulated in their expression. So they're expressed in a certain order. They're expressed in a certain area of the pupa. They're controlled by other proteins. So they're not allowed to just diffuse through the whole organism. But depending on where they are, that then determines what part of the pupa becomes what part. So where's the head? Where's the tail? Where are the wings? Where are the legs? Those things are all controlled by proteins interacting with each other. So I think this is really cool that they actually gone through this and found the different type of genes, and they kind of put them into families here. So where, when and where they act. It's like a choreography, right? Have you ever done a dance or something like that? It's a very carefully controlled process, isn't it? Or a dance. Same thing here. You don't just express all the genes at once and say, well, I hope it forms into something. No, they're carefully controlled. And the proteins interact. So you can see some proteins will activate certain other proteins. Some proteins will go back and either activate or interfere or inhibit the expression of others. So, and I won't read through all of it. But what this can lead to then is you can actually see gradients, right? So here's one cool example of what they call a maternal gene, where they found the RNA is in one place. This is the pupa. So where do you expect to find the protein expressed? Do you find it in the same area as the mRNA? Well, now we got to go back to translation, right? So translation is where we go from RNA to protein. Do you always get protein from RNAs? No, that's a mistake I made as a grad student. You can, just because you express RNA doesn't actually mean you get protein. Remember that whole post-transcriptional regulation I mentioned? There's a whole ton of things that have to happen before you get protein. So just because you get RNA doesn't mean you always get protein expression. But here, what do they find? It's like, oh yeah, if we find, you can actually stain for the RNA. So this is RNA that's been stained. You can actually see where it is in the cells. And then you also see kind of the protein here as well. You see a gradient. So you see more protein over where the RNA is. But then over here, you don't see as much protein. So it's kind of segregated, isn't it, to one side? And this is actually how it forms the gradient or the polarity of the organism. So that this will actually determine which part becomes the head, or what do they call it, posterior and anterior. So it actually sets up the polarity of the organism. And of course, as good scientists, what do? We go through and mutate them, right? Because it's a lot easier to mutate it in fruit flies. They can't really complain than humans, is it? I mean, try to mutate a human. Yeah, let's complain or want more money for it, right? So if you mutate it, what do? You find out, oh man, we can mess these things up. And here, if you get rid of Bitcoin, you actually get a fly with two butts, right? Two posterior ends. Yeah. Okay. So that's what all good scientists do. I mean, how do you figure out what things do sometimes? You mutate it and see what goes wrong. Oh, okay. So this gene must have a role in polarity, something like that. Can you rescue that? That's the term. Sure, I can actually take the RNA and like inject it back into the mutant and get a normal fly from that, okay? So it's through these kind of manipulations that we can learn about the different processes and what these proteins are doing, okay? So I think it's kind of cool to look at something like this and go, okay, well, how do do we get two heads, sir, with a, I don't know if that's a real picture or not, so. Not funny? Genetically modified organisms. Should I give you a trigger warning before that? Are you like GMO? What do you call you guys? Anarchists? What's the word? Anarchists? Is that the word? Okay, whatever. I think it's kind of cool though to see how these different proteins work together. So you can actually see where the RNA is, where the proteins are, and then see how these proteins can actually control the expression of other proteins. So you can see inhibition as well, okay? So you can peruse these. I'm not going to test you on anything on fruit fly development, okay? So there's a whole different class for that. But I want you to think. I want you to think about what this class is about. What does it mean for these things to be regulated? Why do they have to be so carefully regulated? And then, of course, if things go wrong with that, what can happen? What can we learn from that as well? Because making these types of mutations help us see what can happen in the so hopefully that got your brain juices flowing a little bit. Are there any other questions on the course that we didn't go over that are pressing? I'll go back and change the information on my. Yeah, so what we're going to do in lab tomorrow is form our lab group. We have to see who we want to hang out with the rest of the semester. And then we'll start working on the Gen Bio review. We'll talk about safety as well. So it's not really a huge lab, but got to get started with our review, right?